Okay, so this part is going to be from the penicillin case study, mostly talking about polarity, hydrophobic and hydrophilic a little bit, but mostly then focusing on isomers and enantiomers. So to start with, this is review. We've talked about this a little bit before. The big thing here to understand is that whenever something bonds, so if you have a carbon-carbon bond, these have the same electronegativity. So that's something we're going to talk a lot about in class. You'll see that as throughout this unit. So since the electronegativity of carbon is the same as the other carbon, the electrons here are shared. And so we're going to see, and these are shared equally because they are the exact same atom. And so this would be an example of a nonpolar bond because they are the same atom and they're sharing the electrons equally. When we start getting into things like polar covalent, these are sharing electrons, but they might not share them equally. So a good example of this would be like a carbon double bonded to an oxygen. So this is really electronegative. Other examples would be carbon, let's say triple bond to nitrogen. These two elements in particular are very electronegative. So anytime you have these, we have this property of polarity, where the electrons are being pulled in that direction towards those electronegative atoms. So towards oxygen and towards nitrogen. And I'll show you an example of that in another slide here coming up. When we get to a really extreme case, we get what's called ionic bonding, where they're actually not even sharing electrons anymore, they get fully transferred. So if you have like sodium chloride, for example, NaCl, sodium has an electron that gets transferred to the chlorine, because this is a very electronegative, and this one is not. So the electron actually gets transferred fully to chlorine, because remember, they want to have those valence shells filled. They want to have 8 electrons in their outermost rings. So what ends up happening is, since sodium is losing a negative charge, it's subtracting a negative, it becomes positive 1. Chlorine is accepting that negative, so it becomes a negative 1. And so we have an ionic bond that's formed here then. These are like opposite ends of a magnet now, where this is a positive end, this is a negative end, and they will stick to each other really well. We could draw then another, actually sodium here, that's been ionized or become ionic. This will actually be surrounded by sodium ions. And then there will be chlorine ions that fit here. And so like if we look at salt, which is NaCl, under a microscope, you see that it's like very square in its shape. And that's where the squareness comes from, is because it will always like break along one of these bonding points where this structure gets formed from it being positives and negatives. So why is that important? Sorry for the blurriness on the slide. It was just a picture here that got blown up a little bit, but I like this one. Isomers. So isomers are something that have the same formula but different shape or structure. So let's take the molecular formula of C2H6O. So typically we think of that as ethanol, which is the alcohol people drink. You'll notice that there is an OH group right here, which tells you that the polarity of this molecule is in this direction over here, which means this end of the molecule is going to become more negative, and this end is going to be slightly more positive. And so there's all sorts of things that come along with this. So that makes this hydrophilic, and this one we're going to see is hydrophobic. So dimethyl ether, now same formula. So these two are isomers of each other. Same formula, different shape, different structure chemically. Now, the oxygen's in the middle here, and you'll notice that it is sort of a mirror image on both sides. So what's going to happen here is there's going to be some polarity in this direction and some polarity in this direction. Since those are the same, they cancel. And so what ends up happening is this ends up being not polar as a molecule because of that sort of balanced symmetry within the molecule. Even though the carbon-hydrogen bond, even though this is polar and this is polar, they're polar in opposite directions of each other, so they oppose each other. Another good example of this, one that we talk quite a bit about in this class, is carbon dioxide. So in this case, there's polarity. I'm going to draw it with an arrow going this way out and this way out. So this ends up being negative. This ends up being negative. This ends up being really positive. But these will balance. So it's like two people pulling on a rope in opposite directions that are equally strong. And so this ends up being, even though this is a polar bond, this is a polar bond, this molecule is not polar because they oppose each other in opposite directions. Here's some other isomers. I'm not going to ask you to memorize all of these or anything, but you should be able to kind of identify some of them. Like if I asked you, are these isomers? This and this. Maybe I'll do it in blue because this is a green background here. So butane and 2-methylpropane. Are those isomers? Well, you could count them up. There's C1, 2, 3, 4. So there's C4 and H, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So C4H10. And the top one is the same. So yes, those are isomers. We call them chain isomers. I'm not going to get that specific with asking you to identify that part, but similar thing here. Are those the same formula? Yes. Do they look different? Yes. So they're not the same molecule, but2-ene, but1-ene. They might have different functions as a result. Here's another one, but2-ene, cyclobutane. So same formulas, very different looking structures, and again, they'll have different jobs in our body and in biochemistry especially. We're going to spend a little bit of time specifically on these things called stereoisomers. Stereoisomers are ones that are mirror images of each other, and what ends up happening with those is some special circumstances. So I'm going to give you the background here on that. So here's an example. If we had these two molecules, here is a carbon, and then it doesn't really matter what these are, but usually this would represent an oxygen. This one might represent a chlorine. This one might represent a nitrogen, and this one might represent a bromine. Now we look on the other side here. Here's our bromine, the purple, chlorine, nitrogen, and oxygen. And we'll do this in class where we actually build these. So when you look at these, here is a mirror, and both sides of this show that it's basically like facing a mirror. Now, if you tried to take this one and set it right next to this one and make it identical, you could never do that. The red and the blues would never line up with the greens and the purples. There would always be at least one atom out of place with where they're supposed to be. So we call these enantiomers when they're mirror images of each other but have the same formula.